Hello everyone, welcome to Concept and Fact of Life Sciences. Today in this video, we are going to discuss about the largest membrane-bound cell organelle, and it is the nucleus. In our previous video, we have already categorized the cell organelles on the basis of number of membranes they have, and this nucleus, it falls within the double membranous organelles along with the mitochondria and plastid, जिसके बारे में अपन पहले लेक्चर्स के अंदर बात कर चुके हैं. So let's start with the nucleus. Now the true nucleus, it is observed during the interphase. Now what is true nucleus? The nucleus which have the nuclear membrane. In case of the prokaryotes, there is a presence of insipine nucleus, that is the nucleus without the nuclear membrane, while the eukaryotes, they have true nucleus which is having the nuclear membrane. But in case of the eukaryotes, we can only observe this true nucleus during the phase which is called as interphase. Now what is interphase? जानने की कोशिश करते हैं. This diagram, it is representing the cell cycle. Now the cell cycle, what is cell cycle? Cell cycle, it is the dividing and growing cell, passes through the regular sequence of events that leads to the division of cell. So within in cell cycle, we have different phases in the form of G1 phase, S phase, G2 phase, and the M phase. These are the important phases of this cell cycle. Now there is two important phases. It is the S phase and the M phase. Between these two important phases, we have some gaps in the form of G1 and G2. Now during the S phase, there is a process which is called as DNA replication. There is a synthesization of protein which are known as histone protein. So as it is for the synthesis, so during the S phase, there is a synthesis of DNA, there is a synthesization of some histone proteins. Then during this M phase, there is a division. Okay, जो आपका डीएनए रेप्लिकेट हुआ है उसको वापस डिवाइड करना है, सेग्रीगेट कराना है, तो ये एम फेज में होगा. तो यहाँ पे रेप्लिकेशन एंड अगेन देयर इज ए डिवीजन, एंड बिटवीन दीज टू इंपॉर्टेंट फेजेस वी हैव सम गैप्स. ठीक है. Now during this G1S and the G2 phases, the cell prepare themselves for the division. Means all type of proteins which are required for the division are there or not. The DNA is replicated or not. The histone proteins are synthesized or not. There is a check for the error during the replication. So all these processes, they occurs within these G1S and the G2 phases, and together these G1S and the G2 phases, they are called as the interphase. इसी को अपन कहते हैं interphase. That includes the G1S and the G2 phase. This is known as the interphase. Now we can observe the true nucleus during this interphase, क्योंकि जैसे ही cell it will enters into the M phase from the interphase, that is the prophase. During the prophase, there is a disappearance of nuclear membrane. अपन आगे पढ़ेंगे. There is a cyclin BCDK1 complex which phosphorylates the nuclear lamina. There is a disassociation or disassembly of the nuclear membrane, and the nuclear membrane will get disappeared. So ऐसा न्यूक्लियस जिसमें न्यूक्लियर मेम्ब्रेन हो, वो आपको इस इंटरफेज में देखने को मिलेगा. इसलिए अपन कह रहे हैं ट्रू न्यूक्लियस इट इज ऑब्जर्व ड्यूरिंग द इंटरफेज. ड्यूरिंग द फर्स्ट फेज ऑफ द एम और डिवीजन फेज, देयर इज ए डिसअपीयरेंस ऑफ द न्यूक्लियर मेम्ब्रेन. तो आपको डिविजनल फेज के अंदर न्यूक्लियर मेम्ब्रेन देखने को नहीं मिलेगी, और जैसे ही जीवन में आएगी वापस अपन कह सकते हैं देयर इज ए रीअपीयरेंस ऑफ द न्यूक्लियर membrane. So in these phases which are together known as interphase, we can observe the true nucleus. So यहाँ पे अपन cell cycle की regulation की बात नहीं कर रहे हैं. The cell cycle have some checkpoints जो check करते हैं कि cell prepare है या नहीं division के लिए, that is G1S, G2M checkpoint. There is a regulation of cell cycle through cyclin and the CDK, जिसके बारे में आगे अपन separate एक वीडियो बनाएंगे. तो यहाँ पे जस्ट इतना समझना आपने को कि एक जो ट्रू न्यूक्लियस है, सत्य केंद्रक जिसे कहते हैं, ऐसा केंद्रक जिसके चारों केंद्रक झिल्ली उपस्थित है, वो अपने को केवल इंटरफेस के अंदर देखने को मिलता है. नाउ इफ वी टॉक अबाउट द स्ट्रक्चर ऑफ ए न्यूक्लियस, एज वी ऑलरेडी स्टेटेड दैट इट इज ए डबल मेम्ब्रेनस ऑर्गेनल. नाउ दिस इज रिप्रेजेंटिंग द सेल मेम्ब्रेन. We have cytoplasm which remain present within the cell membrane. Within the nucleus, the plasma is known as the nucleoplasm. और जैसा कि अपन ने पहले कहा था, the nucleoplasm and cytoplasm they together known as the protoplasm, जिसे अपन जीव द्रव्य कहते हैं. Now the nucleus they have two membranes. There is a presence of outer membrane. There is a presence of inner nuclear membrane, like the mitochondria and the plastid. Now the nucleus they contains the genetic material, and that genetic material it is known as the chromatin. उसे अपन कहते हैं chromatin. Now within the nucleus or beneath the inner nuclear membrane there is presence of another layer which is known as nuclear lamina. Now this nuclear lamina it is generally made up of the intermediate filaments which are rigid in structure, which is responsible for maintaining the shape of this nucleus. जो इस न्यूक्लियस का जो शेप है उसको बनाए रखती है, कौन बनाए रखती है? न्यूक्लियर लैमिना. Now within the nucleus we have some processes, that is we can say the formation of the mRNA from this DNA. The process is known as transcription, है ना, जिसे अपन कहते हैं ट्रांसक्रिप्शन. So this is all about the structure and some functions of the nucleus. तो बात करते हैं कुछ important nucleus के structural components की और कुछ important functions के बारे में. And one more thing, this nuclear membrane they contain some transporters, and these transporters they are made up of certain proteins which together forms a complex which are known as the nuclear pore complex. Through these NPCs or the nuclear pore complex, there is a exportation and importation of molecules in and out of the nucleoplasm, or we can say through this nuclear pore complex, the nucleus can communicate with the cell cytoplasm. So start करते हैं अपन न्यूक्लियर पोर कॉम्प्लेक्स के साथ ही कि न्यूक्लियर पोर कॉम्प्लेक्स होता क्या है. इट इज वेरी मच इंपॉर्टेंट in relation to the functioning of nucleus. So the nuclear pore complex it is made up of various nucleoporins, है ना. Porins जैसा अपन पहले बात कर चुके हैं, these are the transporters which remain present within the plasma membrane, and the porins which remain present in the nuclear membrane they are called as nucleoporins. Now 30 this type of nucleoporins they together forms a complex, and this complex is known as the nuclear pore complex. Now if we talk about the size of this nuclear pore, जो nucleoporins है उसका size क्या होता है? The average size of this nucleoporine it is 9 nm, that is equivalent to the 90 angstrom. But if we talk about the humans, the size of nuclear pore it is only 5.2 nanometer, that can be 10.7 in case of the xenopus. xenopus के अंदर आपको ये साइज 10.7 देखने को मिल सकती है. तो एवरेज साइज अपन मान के चलते हैं, दैट इज 9 nm. सपोज कीजिए दिस इज रिप्रेजेंटिंग द न्यूक्लियर पोर, एंड द साइज ऑफ दिस न्यूक्लियर पोर इज ओनली 9 nm. But the molecules that is of 50 kilodalton they can easily pass through this nuclear pore. How it is possible? Because we can widen these pores, that is 9 nm, we can widen these pores from 9 nm to 40 nm. And the molecules which plays an important role during this process, these are called as the Ran-GTP, which belongs to the category of small g protein. Now, how there is a functioning of these Ran-GTP during the process of nuclear importation and exportation, इसके बारे में आगे अपन डिस्कस करेंगे. Here we are discussing the structure of nuclear pore complex. Now we have nuclear outer membrane and nuclear inner membrane. Within the inner membrane, we have plasm, which is known as nucleoplasm, जिसे अपन कहते हैं there is a presence of nucleoplasm. And outside the nucleus, we have cytoplasm. So we can say it is a permeability barrier, that is nuclear membrane between the cytoplasm and the nucleoplasm. Now, if we talk about the structure of this nuclear pore complex, it is made up of a central channel, and the size of this central channel, it is 9 nm. This central channel, it remains associated with a cytoplasmic ring, the ring which remain present towards the cytoplasm. It is known as cytoplasmic ring, which includes 8 protein complexes. Now, these cytoplasmic ring, it remain attached to the central channels through some proteins which are known as the spokes. Similarly, this central channel, it remain also associated with the ring which remain present towards the nucleoplasm and called as the nucleoplasmic ring. उसे अपन कह रहे हैं, it is a nucleoplasmic ring through the proteins which are called as the spokes, जिसे अपन कह रहे हैं spokes. Further, the cytoplasmic ring, it associates with some filaments towards the cytoplasm, and these filaments are called as the cytoplasmic filaments, while the filament which remain present or which attaches to the nucleoplasmic ring, these are called as nucleoplasmic filaments. Now, for the molecule to transport from cytoplasm to the nucleoplasm, it is first recognized by the cytoplasmic filament. Then this cytoplasmic ring, it will rotate. Due to the rotation of this cytoplasmic ring, the pore size, it will increase. That is from 9 nm to 40 nm, and there is a transportation of molecule into the nucleoplasm. Similarly, during the exportation of molecule from the nucleoplasm, the molecule, it will get first recognized by these nucleoplasmic filaments, which make conformational change in the nucleoplasmic ring. This nucleoplasmic ring, it will rotate, that will widen this pore, and the molecule will be exported from the nucleoplasm. So in this way, there is a transportation in and out from the nucleus. कैसे होता है, मैकेनिज्म क्या रहती है, उसके बारे में बात करते हैं. So we will take the nuclear transportation. किस तरीके से चीजें न्यूक्लियस के बाहर और अंदर जाती है, कौन कौन सी वो चीजें होती है जो न्यूक्लियस से बाहर एक्सपोर्ट की जाती है और कौन सी चीजें होती है जो इंपोर्ट की जाती है. तो फिर से एक बार थोड़ा सा स्ट्रक्चर समझने की कोशिश करते हैं. वी हैव सेल मेम्ब्रेन, अपने पास में there is a presence of a cell membrane. Within the cell, we have nuclear membrane and nucleus. This fluid which remain present within the cell, it is called as cytoplasm, and within the nucleus we have nucleoplasm, जिसके बारे में अपन पहले बात कर चुके हैं. Now within the nucleus we have nuclear genetic material. अपने पास में न्यूक्लियस का जेनेटिक मटेरियल होता है, and that genetic material, it is known as chromatin. Okay, and within this nucleus we have processes which are known as the transcription. And what is transcription? It is the conversion of this DNA into the mRNA. So DNA से क्या बन जाता है? It's a formation of the mRNA. Now this mRNA, it can be transported out of the nucleus through the nuclear pore complex. Simple. It will first recognized by the nucleoplasmic filament. The nucleoplasmic ring, it will rotate and it will be transported into the cell cytoplasm. So now we have the mRNA within the cell cytoplasm. Now within the cytoplasm we have another cell organelle which is called as the ribosome. So ribosomes, they remain present within the cytoplasm, and the binding of ribosomes to the mRNA, as we all know, it will lead to the synthesization of proteins. That process is called as the translation. Now, in eukaryotes, the transcription, it occurs within the nucleus, while the translation, it occurs within the cell cytoplasm, that lead to the synthesization of a polypeptide. They have NH2 and 1 on one side, they have COH group on the other side. Now, as we all know that within the nucleus, there is a presence of another structure which is called as the nucleolus. That was firstly discovered by the scientist Fontana, that remain present within the nucleoplasm. So we can say this is the portion or the part of nucleoplasm जहाँ पे there is a synthesization of rRNA. यहाँ पे इसे rRNA factory भी कहते हैं. There is synthesization of specific type of RNA that is called as the rRNA. कैसे सिंथेसाइज होता है इसके बारे में अपन मॉलिक्यूलर बायोलॉजी में बात करेंगे. यहाँ सिर्फ इतना समझना है क्या क्या चीजें बाहर एक्सपोर्ट की जाती है, क्या इंपोर्ट की जाती है. सो देयर इज ए rRNA एंड दीज ribosomes, these are basically the ribonucleoproteins. इनको अपन कहते हैं ribonucleoproteins, and these ribosomes, they are made up of rRNA and proteins. ये बने हुए रहते हैं rRNA और प्रोटीन से. सो rRNA तो आपके पास न्यूक्लियस के अंदर है, but you have to transport certain proteins. Now these nucleus, they have cytosolic proteins. In next video अपन बात करेंगे, जब एंडोप्लाज्मिक रेटिकुलम की बात करेंगे, the types of protein on the basis of their site of synthesization. There are two categories of protein. One, we have the cytosolic protein which synthesize within the cytoplasm, जैसे कि अभी अपन ने बात की. And next type of the proteins, they are called as the secretory proteins. उसमें क्या होता है कि जो इनिशियल mRNA है वो साइटोसॉल में आता है, प्रोटीन सिंथेसाइजेशन स्टार्ट होता है, कुछ पॉलीपेप्टाइड्स का फॉर्मेशन होता है, देन दे हैव सर्टेन सिग्नल्स दैट विल ट्रांसपोर्ट देम इनटू ऑनटू द endoplasmic reticulum membrane, rough endoplasmic reticulum membrane, and the synthesization of protein occurs within the endoplasmic reticulum lumen. Then it will be transported to the Golgi, and then Golgi decide their destination. और वो secretory protein काम करते हैं Golgi body के अंदर, endoplasmic reticulum के अंदर, lysosome के अंदर, जो आगे अपन बात करेंगे. But the nucleus, mitochondria, plastid, they have cytosolic proteins. So ऐसे प्रोटीन जो कहाँ सिंथेसाइज हो रहे हैं कंप्लीटली विद इन द साइटोसॉल, उन्हें अपन साइटोसॉलिक प्रोटीन कहते हैं. एंड द न्यूक्लियस, दे हैव साइटोसॉलिक प्रोटीन. नाउ दिस ribosomes, this require proteins along with the rRNA. So this protein, suppose it is a ribosomal protein, and they have, if they have certain signal sequences which are known as the NLS. And what are NLS? NLS, simply these are the nuclear localization signals. These are the nuclear localization signals. Basically, these are the stretch of certain basic amino acids which are rich in lysine and the arginine. So if the protein, they have NLS, that is the nuclear localization signal, then it is recognized by some of the karyopheric, the molecule which helps in the process of nuclear importation, and this protein then will be translocated. Suppose यहाँ से राउंड ले लेते हैं, this protein it will get translocated into the nucleoplasm through the pore complex, which are called as the nuclear pore complex. Similarly, in this transportation against the molecule, it will get first recognized by the cytosolic filament. The cytoplasmic ring rotate, which will wide the pore, and there is an importation of this protein. Now, this protein, which is basically a ribosomal protein, it will then get attached to this rRNA. The binding of this proteins to the rRNA, they will lead to the formation of structure which is called as the ribosomes. Now these ribosomes to get exported from the nucleoplasm. अब इन ribosomes को assembly के बाद कहाँ जाना है? Cytoplasm में जाना है, because the process that is called as translation it occurs within the cytoplasm. इसलिए अब ribosomes का काम नहीं है nucleoplasm के अंदर, they have function within the cytoplasm. So there is a exportation of these ribosomes, and again the exportation is through the nuclear pore complexes. So these ribosomes, they should have some signal sequences which are called as NES, that is nuclear exporting exportation signals. And if they have the NES sequences, then there is a exportins proteins which recognize these ribosomes, and again there is a translocation of these ribosomes from nucleoplasm to the cell cytoplasm. ठीक है, तो इस तरीके से कुछ इंपोर्टेशन एक्सपोर्टेशन प्रोसेस चलता है. थोड़ा डिस्कस कर लेते हैं क्या इंपोर्ट करता है न्यूक्लियस और क्या एक्सपोर्ट करता है, जैसे कि हम बात कर चुके हैं. सो इफ वी टॉक अबाउट द एक्सपोर्टेशन, दैट इज अ मॉलिक्यूल व्हिच इज बी ट्रांसपोर्टेड आउट ऑफ द न्यूक्लियस. तो पहली चीज थी वो क्या थी? mRNA. वी हैव द एक्सपोर्टेशन ऑफ mRNA. द सेकंड थिंग इज द राइबोसोम्स आफ्टर असेंबली. असेंबली के बाद में राइबोसोम्स. सिमिलरली राइबोसोम्स के केसेस में अपन ने क्या पढ़ा? वी हैव टू इंपोर्ट सम प्रोटीन, and the nucleus they have cytosolic protein. So all nuclear protein they are cytosolic, and other type of protein they are called as the secretory proteins, जिसके बारे में आगे अपन बात करेंगे. But the protein which remain present within the nucleus they are cytosolic. Now the proteins which have the NLS sequences, that is nuclear localization signal, which is the stretch of certain basic amino acid, that is lysine and the arginine. It is recognized by the some molecules or import molecules which are known as karyopharines, that will lead to the transportation of these we can say proteins from cytoplasm to the nucleoplasm. So कुछ molecules import होते हैं, कुछ export होते हैं. अब ये इंपोर्ट एक्सपोर्ट मैकेनिज्म किस तरीके से चलती है बात करते हैं उसके बारे में. सो फर्स्ट वी विल टेक द न्यूक्लियर ट्रांसपोर्टेशन मैकेनिज्म, एंड हियर इट इज द इंपोर्टेशन. अपन ने बोर्ड को दो पार्ट्स में डिवाइड कर लिया है. वन इज द न्यूक्लियोप्लाज्म, ये रिप्रेजेंट कर रहा है न्यूक्लियोप्लाज्म को, एंड अदर इज द साइटोप्लाज्म. नाउ एज वी ऑल नो दैट द न्यूक्लियस दे हैव साइटोसॉलिक प्रोटीन, और अपन ने पढ़ा था कि डीएनए तो न्यूक्लियोप्लाज्म में ही होता है, ट्रांसक्रिप्शन भी न्यूक्लियोप्लाज्म में होता है. देन mRNA it is transported into the cytoplasm where there is a binding of ribosome and there is synthesization of some protein. And suppose within the cytoplasm we have a nuclear protein. And how you will recognize that it is a nuclear protein? Because it contains the nuclear localization signals. So if the protein it have the nuclear localization signal, then we can say that it is first recognized by the protein which is known as the importin alpha. So जैसे ही ऐसा कोई प्रोटीन जिसके पास में एनालिस सीक्वेंस है, it will get first recognized by the importin alpha. Then there is a binding of the importin beta that leads to the formation of a ternary complex. Now why it is a ternary complex? because here we are having three components, that is the protein which is to be translocated into the nucleus. We have importin alpha and the importin beta, so it is a ternary complex, simply we can say. Now this importin alpha and importin beta, they together known as the karyopherins. इनको अपन कहते हैं, these are the karyopherins which helps in the nuclear importation process. Now the binding of this importin beta to the protein importin alpha complex, it will lead or it will facilitate the movement of this complex, this ternary complex towards, we can say, towards the nuclear pore complexes. Now here, this complex, this ternary complex, it is first recognized by the cytoplasmic filament of nuclear pore complex. The cytoplasmic ring, it will rotate, it will widen the pore from 9 nm to 40 nm, and then there is a importation of this ternary complex into the nucleoplasm. Now within the nucleoplasm we have Ran-GTP. Ran, as we all know, it is a small g protein. So this Ran-GTP, it will get bind to the importin beta. Now the binding of this Ran-GTP to the importin beta, it will make the conformational change in the importin beta, that it is now not able to recognize this importin alpha, and it will get detached from this ternary complex in the form of importin beta Ran-GTP. In next phase, within the nucleoplasm, we have cellular apoptosis susceptibility protein, that is known as CAS protein, which remain bind to the Ran-GTP, that is a small G protein. Now this binary complex, the complex of two components, it will get first attached to, it will first attaches to the importin alpha. The binding of this Cas Ran-GTP to the importin alpha, it will make the conformational change in the importin alpha in such a way that it is now not able to recognize this nuclear protein which is translocated from cytoplasm to the nucleoplasm. So they will get detached from this प्रोटीन, दिस न्यूक्लियर प्रोटीन, और जो ट्रांसक्रिप्ट है इसकी प्रोटीन की, इट विल गेट फ्री विद इन द न्यूक्लियोप्लाज्म. अब हो सकता है ये एक राइबोसोमल प्रोटीन है, तो ये जाकर के आरआरएनए के साथ अटैच हो जाएगा एंड लीड टू द फॉर्मेशन ऑफ राइबोसोम्स. So in this way there is an importation of molecules from cytoplasm to the nucleoplasm. Now within the nucleoplasm we have importin beta and the alpha. अब इनको वापस से आपको साइटोप्लाज्म में भेजना है so that they can be utilized in the next cycle. So these Ran-GTP Cas importin protein, they will first get recognized by the nucleoplasmic filaments. The nucleoplasmic ring, it will rotate, then they will comes into the cell cytoplasm. Similarly, it will be in the case of importin beta. Again we can say the movement is passive. चाहे आपने अंदर मॉलिक्यूल ट्रांसपोर्ट किया हो, उसमें भी that was triggered by the binding of importin beta, which facilitate the movement of the ternary complex from cytoplasm to the nucleoplasm. Within the nucleoplasm there is a binding of certain Ran-GTP, and again they will get dissociated from the molecule which is transported from outside to inside. Then there is a transportation of these complex back into the cytoplasm, and this all movements they are passive till now, है ना. ये वही process होगा and it will comes into the cell cytoplasm. Now within the cytoplasm, these Ran-GTP, it will get replaced by the Ran-GTP. So Ran-GTP, it will get replaced by the Ran-GTP. Now the binding of Ran-GTP to the Cas and the importin alpha complex makes the conformational change in it, and now this importin alpha and the Cas, they will get dissociated from each other. We can say they will get dissociate. from each other. And this importin alpha, now it is available for next round. Next round means when we have another nuclear protein, or we can say we can clone it, we have another category of nuclear protein, they can recognize these by these importin alpha proteins. Now we have to free this importin beta also. Then again this GTP, sorry, it will replaced by the Ran-GTP. It will make the conformational change in the importin beta. Now it is not remain attached to this Ran-GTP. So it again get free within the cell cytoplasm. It will bind to the importin alpha and the protein complex that lead to the formation of a ternary complex, moves in the nucleus. So in this way, there is a transportation. Now this movement, as we already stated, it is a passive movement. चाहे वो बाइंडिंग हो इंपोर्टेंट की, दैट फैसिलिटेट द मूवमेंट टुवर्ड्स द न्यूक्लियोप्लाज्म, चाहे वो मूवमेंट हो इन मॉलिक्यूल्स का बाहर की तरफ, बट देयर इज ए इन्वेस्टमेंट ऑफ एनर्जी इन द फॉर्म ऑफ जीटीपी. सो इसको पैसिव की जगह एक्टिव भी कंसीडर किया जाता है, क्योंकि यहां पे आप क्या कर रहे हैं, वी आर रिप्लेस्ड टू जीटीपी विद द जीडीपी. नाउ दिस ट्रांसपोर्टेशन, इट इज थ्रू, वी कैन से it is through a nucleocytoplasmic Ran-GTP gradient, है ना. It is through the nucleocytoplasmic Ran-GTP gradient. अभी क्या होता है, that the Ran-GTP concentration, it always remain lower within the cell cytoplasm, while the Ran-GTP concentration, it always remain higher within the nucleoplasm. So there is a movement of this Ran-GTP from its higher concentration to the lower concentration. So this movement, it is due to the nucleocytoplasmic Ran-GTP gradient. That is a passive movement, that is from higher concentration to the lower concentration. Now within the nucleoplasm, we have the higher Ran-GTP concentration. Why? Because the nucleoplasm, we have Ran-GEFs. Now, what are Ran-GEFs? These are the guanine exchange factors. What is the function of these guanine exchange factors? Simple. They convert the Ran-GDP into the Ran-GTP, which keeps the concentration of Ran-GTP higher within the nucleoplasm. That triggers the movement of Ran-GTP from higher to lower concentration. That is for the creation of a nucleocytoplasmic Ran-GTP gradient that triggers this transport from inner side to the outer side. So this is all about the importation mechanism. So आप conclusively क्या कह सकते हैं, ये movement passive है या active है? Although it is a passive movement, but it includes the investment of energy in the form of 2 GDP. So it is a active process. So इसको अपन एक active transportation ही consider करेंगे, but the movement is due to the nucleocytoplasmic Ran-GTP gradient. Next, it is the nuclear transportation mechanism, that is the exportation. Now we will export the molecules from nucleoplasm to the cytoplasm. अपने को क्या करना है, इधर से मूवमेंट करवाना है साइटोप्लाज्म की तरफ. कैसे होगा? Suppose we have nucleolus. Within nucleolus, we have rRNA factory. There is a binding of protein to the rRNA that leads to the formation of ribosome. And these ribosomes, they have some sequences which are known as nuclear exportin signals. Now these nuclear exportin signals, these are first recognized. These are first recognized by the proteins which are called as exportins. ये सबसे पहले एक्सपोर्टिन से एक्सपोर्टिन इनको रिकॉग्नाइज करेगा, है ना. वहां पे क्या कर रहा था, इंपोर्टिन रिकॉग्नाइज कर रहा था, यहाँ पे एक्सपोर्टिन रिकॉग्नाइज करेगा. द बाइंडिंग ऑफ दिस CRM1 टू द NES sequences, it will trigger the binding of a Ran-GTP molecule. Now again this ternary complex, that is Ran-GTP, CRM1 and the ribosome, it will move from nucleoplasm into the सेल साइटोप्लाज्म. ठीक है, इसका मूवमेंट हो जाएगा कहां से, फ्रॉम द न्यूक्लियोप्लाज्म इनटू द cell cytoplasm. Now within the cytoplasm, this GTP, it will get replaced by the Ran-GTP. That will make the conformational change within the CRM1. The CRM1 now not able to recognize the NES sequences, and it will get detached from this ribosome complexes. Now the ribosomes, they are free within the cell cytoplasm, which can take part in the process of translation. Now this Ran-GTP CRM complex, it can again get transported into the nucleoplasm through the NPC, that is the nuclear pore complexes. Now within the nucleoplasm, we have, as we already stated, we have Ran-GEFs. Now these Ran-GEFs, they triggers the replacement of this Ran-GTP with the Ran-GTP. Now these both complexes, that are importin and the Ran-GTP, they get free within the nucleoplasm for the next round of transportation. So इस तरीके से राइबोसोम कुछ बाहर आ जाते हैं. Now in the case of mRNA, although the story is not completely known, but it is supposed that within the nucleus, as you all know, that you have DNA, and the DNA will be converted into the mRNA. Okay? That is through the process of transcription. Now this mRNA, it contains some introns, and it requires some processing. So it is not a mature mRNA. It will convert into a mature mRNA through the process which is known as processing. Now the processing, it includes the 5' capping, 3' polyadenylation, जिसके बारे में आगे अपन molecular biology में बात करेंगे, and it also includes the process which is known as splicing. Now what is splicing? That is the removal of intron and the reunion of the exons, यानी नॉन कोडिंग सीक्वेंसेस को निकालना और कोडिंग सीक्वेंसेस को वापस अटैच करना. दैट प्रोसेस कॉल्ड एज स्प्लाइसिंग. इसके बारे में अपन बात करेंगे अच्छे से मॉलिक्यूलर बायोलॉजी के अंदर. यहां से कितना समझना है कि विद इन द न्यूक्लियस वी हैव ट्रांसक्रिप्शन, वी हैव प्रोसेसिंग ऑफ mRNA. नाउ दिस प्रोसेसिंग process, it recruits some of the proteins in the form of tracks to the mRNA. Then the tracks is recognized by the TAP protein. Then the TAP, they contains the nuclear exportin signals. Now it is recognized by the exportins, which might not be the CRM1, but these are the exportins. Then there is a binding of the Ran-GTP, and it will lead to the transportation of these complexes from nucleoplasm to the cell cytoplasm. Again, this Ran-GTP, it will get replaced by the Ran-GDP. They are now not able to recognize the NES. We can say they will get free from the NES. And again, there is an importation of this importin and the GTP complex. We have Ran-GEF, which exchange this GTP with the GTP, and the exportin and the Ran-GTP get free from each other. So in this way, there is an exportation of molecules from nucleoplasm to the cytoplasm. So this is all about the nuclear transportation. Next अपन बात कर लेते हैं एक important चीज की, जिसे अपन ने कहा था nuclear lamina. Now, as we already stated that we have nuclear outer membrane, we have nuclear inner membrane. Now beneath the inner membrane, we have structure which is known as the nuclear lamina. There is a presence of nuclear lamina. Now this nuclear lamina, as it is made up of some intermediate filaments. Now beneath the plasma membrane, there is a presence of cytoskeleton filament that is called as actin. And actin, we can say it is a flexible protein. while these intermediate filaments, they are rigid protein which are responsible for keeping the structure of nucleus intact. वो एक न्यूक्लियस का स्ट्रक्चर बनाए रखते हैं. नाउ स्ट्रक्चर न्यूक्लियर लैमिना, इट इज द स्ट्रक्चर विद इन द न्यूक्लियर मेम्ब्रेन. जैसा कि अपन ने बताया, it provides strength because it is made up of intermediate filament, provides strength to the nuclear membrane. Now it is made up of various lamines. Now these lamines, they are the intermediate filaments that belongs to the category of type 5 intermediate filament. अपन बात करेंगे, कैटेगराइज करेंगे जब सेल साइटोस्केलेटन पढ़ेंगे. वी हैव डिफरेंट टाइप्स ऑफ intermediate filament, and that these lamines, they belongs to the category of type 5 intermediate filaments. So we have different types of lamines, that is lamines A, B, C, which remain associated with the proteins. So intermediate filament with some proteins, they together forms the न्यूक्लियर लैमिना. ये बनाते हैं न्यूक्लियर लैमिना को. नाउ व्हाट आर द फंक्शन ऑफ दिस न्यूक्लियर लैमिना? फर्स्ट फंक्शन इज it plays an important role in the organization of chromatin. So these lamina, they have docking sites for the attachments of the chromatin and helps in the proper organization of the nuclear genetic material that is known as chromatin. So bind हो सकता है. So it's like some histone, they have affinity for the nuclear lamina. They can get attached with the nuclear lamina, that helps in the proper organization of chromatin within the nucleoplasm. Next thing, they plays an important role in the nuclear membrane disappearance. आगे अपन जब सेल साइकिल पढ़ेंगे उसके अंदर बात करेंगे. There is a complex that is called as cyclin B CDK1. ठीक है, आगे बात करेंगे. CDK, these are the kinases which have ability to carry out the phosphorylation of the nuclear membrane or nuclear lamina. ये क्या करेंगे? It will add the phosphate to the nuclear lamina. The binding of phosphate to the nuclear lamina, as it is a protein, it will make conformational change in the nuclear lamina. The nuclear lamina get depolarized along with the nuclear membrane. That plays an important role in the nuclear membrane. We can say nuclear membrane disappearance during the प्रोफेज स्टेज. जैसा आपने को पता है, प्रोफेज में क्या होता है? न्यूक्लियर मेम्ब्रेन डिसअपियर हो जाती है. क्यों होती है वो डिसअपियर? देयर इज अ रोल ऑफ न्यूक्लियर लैमिना. थर्ड रोल इज द डीएनए रेप्लिकेशन. नाउ द रेप्लिकेशन इज द प्रोसेस वेयर देयर इज अ सिंथेसाइजेशन ऑफ डीएनए फ्रॉम द डीएनए दैट अकर्स ड्यूरिंग द एस फेज ऑफ द सेल साइकिल, जैसा आपने स्टार्टिंग के अंदर बात की थी. सो ड्यूरिंग द प्रोसेस ऑफ डीएनए रेप्लिकेशन, दैट इज द सिंथेसाइजेशन ऑफ DNA from the DNA, there is a phase which is called as elongation phase, and these nuclear lamina, they help in the proper assembly of that elongation complex. So we can say they help in the process of DNA replication also. Now fourth function, it is they also involve in the process of DNA repair. Now during the DNA repair, there is, we can say, there is a breakdown of the two DNA strands, and they will get attached with each other through the process which is called as non-homologous end rejoining method. Now the proteins which help in the process of non-homologous end rejoining, the nuclear lamina, it proliferates the amount of these protein which helps in the process of non-homologous end rejoining. इसके बारे में अपन म्यूटेशंस के अंदर आगे बात करेंगे मॉलिक्यूलर बायोलॉजी में. जस्ट आपको ये समझना है कि न्यूक्लियर लैमिना, दे आल्सो प्लेज एन इंपॉर्टेंट रोल in the DNA repair. So after the repair, to attach the both of the strands with each other, the process is called as NHEJ, that is non-homologous end rejoining, and the protein which are involved in this particular process, these proteins are, we can say, proliferated, or we can say the nuclear membrane lamina, it will increase the amount of that particular protein. So they also helps in the process of DNA repair. So this is all about the structure of the nucleus, some of the function of the nucleus. We are not included here the DNA, or we can say the nuclear genetic material which is called as chromatin. Chromatin के organization, formation of chromosome, इसके ऊपर अपन एक अलग वीडियो लेंगे. Thank you very much.